We're going to make some progress for our BIO410 and BIO510 lab class. Woo! Let's see. Today, I'm sorry, there's no day lecture, and so our goals today are we're going to talk through lab report 2, share the data with you, talk through the data, and then talk through the report. And we should be able to complete it. Except for your questions that you should bring to class, and we should wrap up number 2. Then we're going to talk about lab report 3. And in lab report 3, we're going to do a procedure called immunohistochemistry. And we're going to repeat some of the early experiments that were done in my lab, studying that early start of metamorphosis, the early start of the adult development, adult growth, so formation of the moth. And then we're going to also talk a little bit about lab report 4, because lab report 3 is another package. So we had lab report 1 that was a big long one, and then lab report 2 is a follow-up on that, where we really get to use the tools we built in lab report 1 and go right to some really exciting science, which is to understand more about this prothoracic gland. And my lab has a lot of unpublished data about the prothoracic gland that's very different than any data that other people have collected, because the prothoracic gland has been studied very carefully over 70 years, but with a different perspective than we're bringing with it. And so the second lab report is really straight. We just have one objective. We're going to have one conclusion like that. The third lab report is also, it's really to learn this method, and it's an important method for us to learn as molecular biologists, cell biologists in our course, and but it also will help us build on this project that we're working on. And then the fourth lab report is going to be another longer one. And so we might get setting a little bit of biology of that there. So let's get going on this. First goal is to talk through lab report 2. So here's our data. It's a gel, and here we are using our CHINMO on the prothoracic gland and the CHINMO PCR primers that we made. And on the prothoracic gland, and I'm sure nobody has done this on Earth before, and so it's an interesting, exciting new result, and we got a really interesting result. It's a really important result to me, so I already repeated it and am working on it further, and we can talk some more about that. We're going to be done with it for our course, but know that I'm going to excitedly move on with it. And so we have prothoracic gland tissues that I collected at 12 hours and 24 hours. And remember, the timing is always based on when the animals are put back on food. So they are identified as they molt from the 4th to the 5th growth stage. And when they're molting, they can't eat. So they're temporarily removed from food, and they complete the molt, and they all are hungry and ready to go, but they don't have food. And the start of metamorphosis is dependent on nutrient input, and so we put them back on food all at the same time, and we get a really synchronous start. That means the animals are all developing at the same rate, and that's really significant. That makes results reproducible, right? And really convincing. And we can do lots of experiments and get a lot of convincing data. And we're doing that here, as you are going to see. All right, so here's what we have. We have 4 groups, and each group ran the same set of PCR tubes. Each group set up 3 PCR tubes, and they go, the first tube is an actin positive control. And so we'll look at the 4 groups here. There's actin, worked great, actin worked great, actin worked great, actin worked great. Okay? And then the next tubes are going to be our experimental tubes with our CHINMO primers. Before I talk about that, I just want to talk through the groups. The first 2 groups tested, really repeated the same results, and they tested the prothoracic gland at 12 hours. And there we expected, just based on results from my lab, that we'd expect the CHINMO to be expressed under the model that Jim and Lynn published in Fruit Flies, and then at 24 hours. And at 24 hours, we'd expect Chinmo to be off. And one reason we say that is because we see in my lab expression of BR is off here and starts to be expressed here. So that's what we're checking out. Then the 4 groups. And so here are the people, I'll just read the names of the people who are in the different groups. So this first group was Mariah, Zach, and Godwin, group 1. Group 2 was Ellie, Tiba, and Amol. Group 3 was Joe, Zofia, Jeremy, and Amira. And group 4 was Hannah, Julia, and Max. Okay? And so then when I added the enzyme, so you guys set up the tubes, and at the end of what you set up, there should have been 49.5 microliters in each tube, and then I add half a microliter of the TAC DNA polymerase to do the copying during the PCR process. So that's the enzyme that does the DNA copying, and it's added right at the end. And I have to add it under kind of special conditions, and so that's why I add it, is just to get everything started, and it's just a tiny amount of material. I use a different pipetter with different tips to add such a small volume. So at the start, we should have 49.5 microliters, and I could set a pipetter at 49.5 microliters and just pick up each of these volumes with a different tip each time, because I don't want to cross-contaminate our samples, and just double check the volume. And I was worried about the first group's samples because when I checked that, I saw that the first tube had only about 40 microliters in it, and the third tube only had about 30 microliters in it. And so I was really concerned that they were not going to work like that, just based on the volume. All these other tubes had really similar volumes, and it seemed to be very close to the expected volume like that. And so we started out, even before the PCR reaction were done, concerns about this. Okay? All right. So now let's move forward. We have 2 Chinmo primer pairs that we have tested and seem to work well, and we like to use the 2 because it's nice to have 2 independent tests of the same thing, and you'll see there's reasons for that. And then the other thing to say is that the 2 primers are designed against different parts of the gene, different exon spanning primers, and so that could be interesting because it can give us some different somewhat different results based on something called alternative splicing. It could just happen. But anyway, the first primer is we just called J. I think it's called JL, like that. And so for JL, our prediction is that we would expect to see a product in 12 hours and not at 24 hours. And so here's JL. First group, maybe a little product. Second group, lots of product. Third group, no product. Fourth group, maybe a little product. And I think that fits our expectations pretty well, in that I was concerned about how things got set up in the first group. This tube had the right volume, but because the other samples were, the kind of wrong volumes, just overall concerned about this group's results. And that's understandable, and we'll talk through that and ask about questions and stuff. And the concern would be pipetting errors, right? We're beginning pipetters, even the experienced students, and we really need to help each other and really practice that. Then second group, lots of this JL product. Then third group, none. The fourth group, maybe a little bit. There's that mark there, and it could be a little bit. Okay? I should have said at the outset, this lower fuzzy band, just my lab that likes to call it a primer cloud. And that's because lots of primers, there's just a lot of primers used in the reaction to help drive the reaction forward. We have a great molar excess of primers, 50 times more primers than necessary, because we want the reaction to go very fast. And so we want the primers to stick to their targets immediately when we cool the samples down in the PCR instrument. And so that's a primer cloud, and we see it, and we call it a cloud because it's getting fuzzy because the primers are so short, they're diffusing in every direction as we're running the gel. And they started out up here, and the samples were being pulled down. And so it's being pulled through the gel by electricity, but at the same time, just natural diffusion processes are causing it to spread out pretty quickly. The larger products are also spreading out like that, but because they're longer pieces of DNA products, they don't spread out as quickly. If we let this gel run further, the primer clouds actually disappear. I only run these gels really fast for 20 minutes, like that. So we always see these primer clouds. And going back here, you can see, really consistent primer clouds here, and then, some different amounts here, maybe in the first sample, maybe a little bit different like that. But so that's where we're at on that. So I think the JL primers worked as expected, and I think we see gene expression of our Chinmo, Manduka Chinmo, at 12 hours, and then little or no at 24 hours. Then next one is primer we called Z, and it's called ZS, and we'd expect these to be really similar results. And so first group here, just a little bit of product there, more product here. And then third group, no product. Fourth group, a little bit of product. And so I think really sensible results. Concerned about the volume here. We get very little product here. I'm surprised we got any. PCR is pretty fussy, and but we seem to have got a little product here. And then strong product here, and no product here, and a little product here, and so that's where we're at on that. And I think our results are really, significant first results. It looks like, just based on these very preliminary first time in the world experiment, it looks like, Chinmo is likely expressed at 12 hours and down at 24 hours. And I've repeated this with these same samples myself, and I also collected more samples. I've also collected samples at 48 hours. And what I actually got for this is when I repeated these experiments, I saw some expression at 24 hours, and then I have some other samples I collected at 24 hours, and I saw kind of considerable expression at 24 hours, but none at 48 hours. I also just collected at 72 hours, and that's to be expected. When gene expression decreases, it sometimes can take a while for the RNAs to completely disappear. And so I think things are looking good, and I'm really excited about this because there's no reason for anybody to think this gland would be expressing Chinmo or BR. And so far, it looks like it's a consistent expression with Jim and Lynn's model, where we see an early expression of Chinmo with no BR, and then later, as Chinmo drops away, we see BR increasing. And again, the BR experiments were done by students in my lab, myself, in the last couple of years. So we already have that data like that. And so that's our results. And so that would go into your results sections. I will print out that and have that as a handout. That data is a handout, and you'll want to label it. And you'll want to label it and then write about everybody's results, and then your group's result, and then your own result, right? How did the tube go for you? And see if you can come up with explanations why the results worked or didn't work. And for that would fit into the discussion section. And in this case, the discussion section reflects on our experiments. They were very brief, right, like that. And but I think we got some good results, and we got some mixed results, and so you would write about that. And then this discussion section would be forward-looking. There are going to be a lot more experiments done. And so here's some things you could put in the discussion of your lab number 2, and that is, we want to look at BR expression. It's already been done, but now we want to look at it in the exact same samples. And we want to look at BR expression that goes down, up as Chinmo goes down. Another thing is that we want to do something I want to do something that's called a time course. Really common experiment. And I think that the action in starting metamorphosis is at 20 hours after the animals are put back on food. That's based on the timing of the start of the adult cell proliferation. And so we've collected an earlier time point, later time point. A time course is to just take consistent time points, like let's say every 8 hours during this period. So the animals could go back on food, and that would be 0 hours, and the graph would be 0 hours, and then 8 and 16 and 24 and 32, I hope that's right, and 40 and 48 and maybe another one like that. So you'd have a half a dozen or so time points, and the expectation is that for each one we'd see real consistent chinmo expression, and then maybe falling down like that to 0. Okay, and then we'd see a real consistent BR expression that would look something like this, kind of a pretty close to a mirror image of that for BR and chinmo, and this would be a PCR expression. Now, to get really nice data, instead of doing this regular PCR that we've done, scientists and we would need to do something, I need to do something called qPCR, quantitative PCR, where we can actually get relative numbers. And we'll have a student presentation at the end of the semester. USM just has one qPCR instrument because they're pretty expensive, 20 grand or so, and or more, and ours is broken, and we've decided that it's not worth fixing, and Rachel Lastly Rasher has some line on some money to buy one, a new one, but it's not here yet, and I don't think it'll be here yet this semester. There's another qPCR instrument in the building because there's some scientists for this new thing called the Rue Institute that's going to be in Portland that are working in the building till their labs are set up, and there's one in the building, but it's just a little bit complicated for me to get access to use it. And so I don't know that we'll be able to do qPCR with these samples yet, but that's on the path, and we'll definitely learn about qPCR because that's the publishable type of data where you can really get qualitative numbers on these and really see these kind of changes that we can't see just with the just with those photos. Okay, so then next thing to say about our lab report is we've already talked about a title, we talked about an objective, and we just have one objective, and we're just going to have one conclusion, and the conclusion is about expression of chinmo in the PG at these time points at 12 hours and 24 hours, like that. Okay, and that's that, and then this discussion. So remember, we have just a straightforward title, straightforward objective. The background is mostly C lab report number 1. And we have some added information about the prothoracic gland because that's new to us in the background. And then the methods are all just C number 1, like that, except for the dissection of the gland. And, I did that, but you could have a little mention of the dissection of the gland like that, stage gland, and then C number 1 for the method. The method is PCR, like that. Then we have a result, and that's described, and we'll have a handout for that, and then on to a conclusion, and then a discussion. And we'll want to talk more about the discussion because it's a discussion in class on Thursday, and make sure that we get some good ideas about that. So that's that, and that's where we're at on our second lab report. Now on to talking about some of our third lab report. And for our third lab report, we're going to look at this method called immunohistochemistry. And there was a student scheduled to give a talk on this last Thursday. That student has dropped the course, and I could talk about it some. And immunohistochemistry is a strategy where scientists want to be able to identify target molecules, and they want to be able to identify any target molecule. And a common way to do that is to take advantage of the immune system. Our body and other animal bodies, common animals that are used to help in this research would include rabbits, rats, mice, donkeys, goats. Those are some classic ones. And animals that would be living on a farm and then being used in scientific experiments. And what's done is, we have a rat on a body, so we'll just pick a rat. So just imagine a rat, and it's living in a cage and out on a farm. And like a farm that's like a laboratory. And there's other kinds of animals living there too. And what scientists do is they take lots of this molecule of interest and they inject it into the animal. And the animal recognizes it as foreign. And it's especially true because the scientists mix some other chemicals into this injection that help the animal's immune system be activated. The molecules include some molecules that are from bacteria that commonly cause infection. And it's not an infection because there's no living bacteria or even bacterial cells. It's just some molecules from bacterial coats that would really cause the immune system to be activated. And then this foreign molecule of our choice, like that. And the animal makes an immune response, and immune response include activating some cells called B cells that are going to produce some proteins that get released that are called antibodies. So antibodies are proteins similar to, hemoglobin's a protein, and insulin's a protein, and FOX is a protein, and the PDGF receptor and all the other receptors are proteins. So there must be an antibody gene, and it gets activated in these B cells, and the function of these B cells is to release this antibody, and these antibodies will stick to the target molecule. And they function as a sort of flag to help the rest of the immune system identify this as foreign. And they're very specific, and they're very high affinity. So it's a really great tool, and we can make an antibody really against any molecule by doing this strategy. Okay? And so scientists have done that, and lots of times these antibodies are for sale. Companies sell these. And for our lab report 3, we're going to use some antibodies that are for sale. And they're going to be antibodies that recognize the cell cycle. And that's chapter 18 in our lecture course, and we haven't gotten quite there yet. But the cell cycle is something you learned about in intro biology, because the cell cycle is true for all eukaryotes, different in bacteria, but true in all eukaryotes, all plants and animal cells, all the cells in our body, and single-celled eukaryotes, like the yeast that causes bread to rise, which is where the research was first done. And so we have M phase when the cell actually divides, then we have G1, S, and G2. And we have store-bought antibodies that recognize S and M. So any cells that are in S and M would have molecules in it that these antibodies would stick to. And for S phase, we have an antibody that recognizes something called BRDU. And it's a molecule that stands for bromodeoxyuridine. And so this is some background information for lab report 3 that's going to be about immunohistochemistry, and we'll need to add more keywords to the title. One of them will be Manduca, like that, okay? Like we do, okay? So bromodeoxyuridine is a nucleotide. It's not a natural nucleotide. The natural nucleotides are A, G, C, T in DNA, and then A, G, C, U in RNA. And U is uridine. So this is deoxyuridine. It's this nucleotide that has had a bromine group added to it. So a covalent bonded to it. And when that is in a cell, the cell recognizes it as a T. And so if a cell is in S phase, it's synthesizing DNA. It's copying its DNA. It's replicating its DNA. And if we take living cells and add BRDU, the BRDU will get incorporated into the DNA. And it will get incorporated everywhere there's a T, or most everywhere there's a T, there'll be a BRDU. And we have an antibody to it. And so the strategy is to take some living cells, add this BRDU, and for any cell that's in S phase, it will incorporate BRDU into the DNA, and we'll be able to detect it afterwards with this antibody staining, and we'll be able to detect it under a microscope. Okay, and so the BRDU is said to be an S phase marker. It will help us stain any cell that's in S phase. And if you think about it, this my research is that the start of metamorphosis at about 20 hours involves the moth cells begin to grow, and the way we see that is they go through the cell cycle. So there wouldn't be any staining before 20 hours, and after 20 hours, there's lots of staining in these patches of cells that are going to grow quickly to make the adult. Then we have a separate antibody that's an M phase marker, and that antibody recognizes a modified form of histones that we're learning about in the lecture course. It recognizes a phosphorylated form of histone, and it's the H3 histone. So there are five different types of histones, and this antibody recognizes phosphorylated form of histone. The histones are present all the time, but they're only phosphorylated during M phase, and the phosphorylation causes them to fold differently, and the DNA or chromosomes condenses, and that's a famous part of M phase. Is that most of the time in these other parts of the cell cycle and the cell's life, the DNA is spread out in the nucleus and it's not visible. But you can do a karyotype, you can see chromosomes in a cell and line them up like this, and like we could see chromosomal translocations under a microscope and so forth. Those are only detected when cells are in M phase. And they're in M phase because this is a histone. Histone is get phosphorylated. So this phosphohistone H3 antibody is sold, and it's used again by many scientists to study cells that are going through the cell cycle, and it is an M phase marker. And scientists like us like both an M phase marker and S phase marker to provide independent confirmation. So let me just show you what that looks like. So we'll go to slide, and we're going to stain cells that start to produce the adult eye. And so these are good cells to look at because the larva, the caterpillar, has an eye, and it's composed of little units called larva stomatum, and that's singular, stomatum, and plural is stomata. And so there's one, and it has 6. 1, 2, 3, 4, 5, 6. So it has 6 isolated eyes. Each one has a little lens, and you can actually see it on the outside of the animal, and a little set of light-sensitive photoreceptor neurons, that have a optic nerve that goes to the little tiny caterpillar brain. The adult has a great big eye, and it's a new eye. I put this fruit fly on here just to show you how big it is. So just a scattering of cells are going to make an organ that's way bigger than a fruit fly really fast. Because from the start of metamorphosis at about 20 hours until the animal forms a pupa and sheds its skin as a pupa, there's a lot of proliferation because most of the eye grows by the time the animal forms a pupa. And the eye is all folded up inside the caterpillar's head, and like a sock that's inside out. It's really strange. Insect metamorphosis is really a strange thing. It's life on Earth, but it seems like life from another planet. The cells that make the adult eye, so those mouth precursor cells that we're really interested in, are located here, just right next to the larval eye in a little stripe. And so those are the cells we're going to stain. remember the body of a caterpillar is kind of hollow, like our chest cavity, and not solid. And so for the dissection here, we actually can just cut a piece of tissue out that's pretty much the same as this, just a little patch of tissue. And this head is called a head capsule. It's really hard exoskeleton or cuticle. And we just cut that out carefully and stain that and look at it under a microscope by looking at the inside. One way to think about it is the head capsule is a little bit like if you were wearing like a football helmet or a bike helmet over your head, and we're just going to cut the helmet off. And what we're interested in is the lining, the inner lining of that helmet, because that's where these cells are located. And so we just cut that out, lift it off, stain it, and look at that inside under a microscope. Okay? And so I made this a while ago, just a cartoon, and it shows some stages. And we're looking down at an animal's head. So here's a caterpillar's body, and then here's its head, and it's just showing the left side of the body. So here's the right side of the body. It would be a bilateral organism like that. And this is the left mandible, chewing part like that. So this is its mouth, and this is its brain, and then this is the optic nerve going out to those little larval units. And just above, in front of them, this light yellow is the lining or the actual epidermal cell. The green is the non-living, hard, secreted exoskeleton. Okay, and so here's what it looks like. The cells start to divide, and they divide really fast in a stripe. The stripe, we're just looking at a cross-section of it. And the stripe grows really fast, and so these cells quickly start to fold in because we're getting lots and lots of cells. And they fold in more, and as the animal's still a larva, the eyes start to develop further, and our light units start to get developed. Those are called ommatidia. And this is going to be a multifaceted compound eye like you would see in a housefly, or maybe you'd be more familiar with the giant eyes of a dragonfly. And so our moth has these great big eyes with like 10,000 mini eye units. The caterpillar just has 6 for each side of its head. The moth has 10,000. So they can see in a really pretty dramatically improved way from the caterpillar. And you can see the new nerves are growing in, and I'm showing that the larval brain grows a lot. And so my lab's also studied the brain, because the moth brain and the larval brain are the same brain, but the moth brain is growing a lot. And so I'm trying to pick that there. Then, it's growing, folding in like this. It's inside the head capsule, and when the animal pupates, it folds out. And it has some more growing to do, and some differentiation to form the units, but a lot of growth has occurred, and it's hidden inside the caterpillar's head. The caterpillar's head is hollow. And not a big brain in the caterpillar's head capsule. It has some muscles for chomping on stuff, and so just to give you a little visual for that. And then this is a little bit what the results look like. So here's that larval picture again, and then here's the cartoon drawing of it. And this is about the size of the piece that gets cut out for dissection. That's one of the mandibles. You don't cut that off. Whoop, don't cut that off. It looks like that. So cut up that piece, stain it, and then look at it under a microscope. And this is what it's going to look like under a microscope, a little bit early hours and later hours. And I say metamorphosis starts at 20 hours because that's about, it is right after that when we start to see these markers start to be activated. And they get activated slowly, a little bit like that, but we start things happening, and by 40 hours we've seen this. And so each of these dots is actually the nucleus of a cell in this stripe. And the cells are stained with this antibody, and it shows that they are either, in this case, in S phase, because we use the BRDU labeling, or in M phase. And see, the stripe is here, the cells are here, but they're not going through the cell cycle yet. They're just about to start. Okay, and These just shows those individual larval eye units like that. See how that's what we'll see. So we'll be able to use those as landmarks under the microscope, and we'll be looking at this region right next to them to see this staining like that, okay? And we'll use this immunohistochemistry to stain the tissues, and that's what that looks like, okay? And so this just shows that a lot of cells are stained with the BREU antibody. They're in S phase, and a smaller number of cells are stained with phosphohistone H3 antibody that are in M phase. And that's just because this BREU S phase involves an incubation period where the tissues are dissected and then the living tissues are incubated while alive in this BREU. So there's a longer labeling period, and that's why more cells are stained. All the cells in this are going through proliferation. We're only staining a subset of them because some of the cells are in G1 or G2, S, or M. And so here we're staining the cells that were in S. Here we're staining the cells were in M. And there are additional cells like that. Okay? So we're just repeating this result and for lab 3. And so a big thing for lab 3 will be to focus on learning this immunohistochemistry method. And it involves dissection and then this staining. Staining with these antibodies involves several incubations that take overnight incubations. So we'll try to compress that all together. Here's just another picture of that result. And I just wanted to show you this. This is an animal that we fed it, and then we took it away from food, and then we refed it. And the animal got confused, and when it molted, and from the 5th instar, 5th growth period, to form a pupa, this animal was confused, and it molted as a hybrid, an intermediate between a caterpillar and a pupa. And the pupa is brown, caterpillar is green, and so you can see that here, that these cells thought, oh yeah, we're on our way to make a pupa, and the other cells thought we're going to make a caterpillar. And we published this a long time ago. It's really pretty because there are these patches of brown on these defective intermediate animals, and the patches are everywhere that there were adult precursors. And it shows that the adult precursor to grow, and also they commit. They're like, yeah, we're making a pupa because we're on our way to making a moth. So just show you that biology. There's a lot of biology that my lab has published on this topic. This just shows a different kind of view. It uses a fluorescent microscope, and we'll talk about that some in class. And so this just shows one of those larval eye units, a larval stomata. This is one of them. And there's another one down here, and there'd be another one up here. And then this is where the stripe is. And you can see that when we just stain this, these are the nucleus or nuclei of the cells, because this red stain stains the DNA. And so it just shows us where every nucleus is. And it shows us that surrounding this stripe, the cells are big, and along this stripe, the cells are small. And we'll talk about that some in class. These smaller cells are the adult precursor cells, and they're diploid. The surrounding cells are giant cells, and they have many, many copies of each of the parental genomes. They're said to be polyploid. And so you can see that these cells are already specialized cells. They're really separate and different from the caterpillar. And we see that everywhere the adult starts to grow, that they're playing a role in the caterpillar's body, but they already know that they're going to play another role in the animal's life. They're going to play a role of building the adult. So there's just a lot of fascinating biology. Here's part of it. These cells are all epidermal cells. They are skin cells, and they secrete the exoskeleton. And they express the genes that make the extra material for the exoskeleton. And This is a kind of experiment we'll talk about, staining experiment that looks kind of gross, but it's called an in situ hybridization. And we're studying expression of a caterpillar gene that codes for the green pigment. And you can see at this early time, all these cells are dark because they're all expressing the RNA, and we're detecting the RNA for this gene, including these genes are expressing it. So the adult precursor cells act like skin cells as a caterpillar, and then that RNA starts to disappear from this patch, but stays present in the other regions. Because again, the dark staining is staining for the presence of the RNA using this in situ hybridization method. And so you can see these adult cells are surrounded by cells that are still larval cells. And I want to show you that because we'll talk about that some for the rest of our semester, that there's a challenge for us to study CHINMO expression in these other tissues that we're interested in. We're able to study it pretty easily in the epidermis. We're able to study it pretty easily in the prothoracic gland, but it's harder to study in these other tissues. So that's just a little intro to that. And this just shows the timing of the BR expression, and so this is PCR, and this is this qPCR, so publishable PCR. They're just three different versions of the BR gene. They all seem to be expressed in the same way. We start to see their expression at 18 hours, and it goes up like that. And then these are under some different conditions with different hormone mimics, and we're not going to talk about that. So this is just their natural increase. And this is the data that we'll repeat with I'll repeat with the CHINMO samples we've collected, and lots more of them. And we think we'll see CHINMO expression here and then go down. So as CHINMO goes down, we're predicting we'll see BR going up, and we have our first hint of that already with your results. This is one of those in situ hybridizations with BR, showing the RNA in the tissue, and you can see the dark RNA in this case. In the previous slide, it was a caterpillar gene, and it was expressed everywhere, so everywhere was dark, and then it disappeared from the stripe. In this case, the BR gene is not expressed anywhere, and it starts to be expressed in adult precursor cells. So really satisfying combination of PCR and in situ hybridization to say we see expression, and we see expression in these cells here. Okay, so I'm going to stop here for today, and we'll pick it up in class. So I'm going to take notes on this so that you can insert those into your notebook. As we complete Lab Report 2, we start to work toward Lab Report 3, and we'll start also learning about Lab Report 4, which I don't think we've said anything about yet. Okay, so let's stop here, and great. Sorry about the snow day.